Hi all, welcome back to Bloom with Bio. So today we are going to learn the next chapter in human physiology, and that is the body fluids and circulations. So what are body fluids? Body fluids are blood and lymph which are present in our body, and the major body fluid in our body is the blood. So we will be learning in very detail about blood in this chapter, and also we will be learning about lymph. So what is blood? Blood is a fluid connective tissue, or it is also called as the specialized connective tissue. And this blood has many compositions or components in it. So now we are going to learn the composition of blood. So you can see blood is made up of mainly two components. One is the fluid part and the other one is the solid part. The fluid part is called as the plasma and the fluid part is called as the formed elements. So the plasma constitutes 55% of blood volume and formed elements constitutes 45% of the blood volume. Now, formed elements are of different types. One is RBC, the other one is WBC and the platelets. Now coming to plasma. Plasma is constituted by 90 to 92% of water. 6 to 8% of plasma is proteins. So what are the plasma proteins here? One is fibrinogen. This fibrinogen helps in blood coagulation or blood clotting. Then we have globulin. Globulin helps in defense mechanism. Then we have the albumin. Albumin helps in maintaining osmotic balance of our body. So in addition to plasma proteins and water, this plasma is also having certain minerals like sodium, calcium, magnesium, bicarbonate, chlorine, etc. And there is glucose, amino acids, lipids also present in the blood, as these are absorbed from the digested food. Now, in addition to that, plasma also contains hormones. It also contains the waste like urea and the respiratory gases like carbon dioxide and oxygen also. So these all substances are present in the plasma. Serum is nothing but the plasma without clotting factors. So we can write plasma minus clotting factors is serum. Now tell me what is plasma. So plasma can also be written as blood without formed elements. So blood minus formed elements are the plasma. Now what are formed elements? We already told formed elements are RBC, WBC and platelets. And what are these platelets? Platelets are the clotting factors. So can we write formed elements as RBC, WBC and platelets? These platelets are the clotting factors. And these RBCs and WBCs are the blood cells. So how can we write serum in the terms of blood? So serum is blood minus blood cells plus clotting factors. Now let us see what are the formed elements. So formed elements include RBC, WBC and platelets. RBCs are also called as the erythrocytes. WBCs are also called as leukocytes. And platelets are also called as the thrombocytes. So RBCs, the red blood cells, are also called as erythrocytes. WBCs, the white blood cells, which are also called as leukocytes, and platelets, the thrombocytes. So first we will see what is RBC. So RBC is nothing but the red blood cells. we already told that it is also called as the erythrocytes. These are the most abundant type of blood cells present in our body. So what is the RBC count in our body? It is like 5 to 5.5 million RBCs are present in per mm cube of blood. So in 1 mm cube of blood, what is present? 5 to 5.5 million RBCs are present. And the lifespan of RBC is like 120 days. After which what will happen? This will go to spleen and it will get destroyed, and spleen is known as the graveyard of RBC. Now we'll see shape and structure of RBC. RBCs are discoid in shape. So they have discoid in shape and they are biconcave. So what does that mean? So if this is an RBC molecule, it will be having a discoid shape, or like it will be like this. It will be a discoid shape. So why is it? Because it do not have a nucleus. That is it is enucleated. That is, it is devoid of nucleus. So in the early stage, when these RBCs were produced, they had nucleus. But after maturation, these RBCs will lose their nucleus so that they can do their functions. That the function is to transport oxygen. For transport of oxygen, RBCs have a specialized pigment present in their body. And the specialized pigment which is present in their body that helps in transport of oxygen is the hemoglobin. This is an iron containing pigment, or this is an iron containing red color pigment which helps in the transport of gases. So it is also called as the respiratory pigment. Now coming to WBC. So WBCs are white blood cells. They are also called as leukocytes. So these WBCs do not have hemoglobin, so they are colorless. So in 1 mm cube of blood, you can find 6,000 to 8,000 WBCs. So these WBCs have nucleus, that they are nucleated. And their nucleus shape and size differs in different type of RBC. So it is different in different types of WBCs. These WBCs are short-lived, that is their lifespan is just 2 to 3 days. Now let us see different types of WBCs. These WBCs are of two types. One is granulocyte and the other one is agranulocyte. So the granulocytes are having granules present in their cell, and the agranulocytes do not have granules. There are three types of granulocytes. One is neutrophils, then the basophils, then the eosinophils. Coming to agranulocytes, two types of agranulocytes are there. One is monocytes and the other one is lymphocytes. Neutrophils constitutes 60 to 65% of the total WBC. So these are the most abundant type of WBCs present. Now basophils are the least abundant type of WBCs present, and they are like about 0.5 to 1% of the total WBC count. Coming to eosinophil, 2 to 3% of the total WBC is eosinophil. Then 6 to 8% of total WBC is monocytes. The second most abundant type of WBC is lymphocyte, that is 20%. These lymphocytes are of two types. One is T lymphocytes and the other one is B lymphocytes. Now we will see detail about these. So first type of WBC is neutrophil. So these are the most abundant type of WBC, that is 60 to 65% of the total WBC count consists of these neutrophils. And these are the principal phagocytic cells. That is, if some infections or something happens in our body or some pathogens come in our body, these neutrophils are the first person to reach there. They have another name, that is PMNL, that is polymorphonuclear leukocyte. Why are they called so? Why are they called as PMNL? Now let us see structure of neutrophils. So these neutrophils are principal phagocytic cells and they are granulocytes, right? So now let us see the structure of neutrophils. So neutrophils are granulocytes. So we can have a lot of granules in that cell. And these neutrophils are having 2 to 5 lobed nucleus like this. So they will be having 2 to 5 lobed nucleus. So since they have different morphology of nucleus, they are called the polymorphonuclear leukocytes, PMNL. Next we have the basophils. They are the least abundant type of WBCs present, that is only about 0.1 to 1% of WBC constitutes the basophil or is constituted by the basophils. What do? They respond to inflammatory responses. So they respond to the inflammatory responses and they have a S-shaped nucleus. So how do they look like? So we can draw a basophil like this and they have a S-shaped nucleus. And these are granulocytes, so their cell will be having granules. Now this basophil secretes serotonin, histamine and heparin. So heparin is an anticoagulant. So it prevents, so it's an anticoagulant, it prevents clotting of blood. So it prevents blood clotting. Now what is the function of serotonin? So serotonin is a vasoconstrictor. So what does it do? And heparin is a vasodilator. Now let us see what is vasoconstrictor and what is vasodilator. So vasoconstrictor is, this is a normal blood vessel. Okay. So if vasoconstrictor is present, it will constrict the blood vessels. It will constrict the blood vessels. Now what is vasodilator then? So if this is a normal blood vessel, it will dilate the blood vessels. Okay. So you understood what is vasoconstrictor and vasodilator. So the next type of WBC is eosinophils. So these eosinophils are anti-allergic. So what does that mean, anti-allergic? They will respond to the allergic reactions. So this eosinophils constitutes 2 to 3% of the total WBC count. Their nucleus is bilobed nucleus. So how does they look like? So this is eosinophil and they have granules present and they have a bilobed nucleus like this. And next we have the monocytes. Monocytes constitute 6 to 8% of the total WBCs. These are the largest WBCs present. Okay, so these are the largest WBCs and their nucleus is bean-shaped nucleus. So how does they look like? Monocytes are agranulocytes, so they don't have granules and they have a bean-shaped nucleus. They are also phagocytic cells. Okay, so once these monocytes respond to an infection, now these WBCs along with the plasma will go to the tissue and these WBCs will engulf that pathogen. But after which these WBCs cannot come back to the blood. So what happens? These WBCs will differentiate into the macrophages and they will stay in the tissues and do their phagocytic functions. Next is the lymphocytes. Lymphocytes constitutes 20 to 25% of the total WBC count. And we already told that these lymphocytes are of two types. One is T lymphocytes and the other one is B lymphocytes. B lymphocytes and T lymphocytes are also different types. These T lymphocytes are like helper T cells, killer T cells, suppressor T cells, memory T cells. So coming to the B lymphocytes, two types of B lymphocytes are there. They are plasma B cells and the memory B cells. These plasma B cells are the type of B cells which will produce the antibodies. These lymphocytes provide immune response to our body. So what type of immune responses they are providing? B lymphocytes will be giving antibody mediated immunity. And the T lymphocytes will be providing cell mediated immunity. So they have a large round nucleus and they don't have granules in the cells as they are agranulocytes. So the next formed element is platelets. So these platelets are also called as the thrombocytes. So there are 1.5 to 3.5 lakh WBCs per mm cube of blood. And these are formed from the megakaryocyte of bone marrow. Not only these platelets, all the type of blood cells, that is RBC, WBC, platelets, all the type of blood cells are produced from the bone marrow. Now let us see how blood is getting coagulated in our body. That is blood coagulation, or it is also called as clotting of blood. Imagine there is a small cut in my hand. So what will happen? Blood will be lost, right? But after some time, what happens? The bleeding will stop. So why is it happening? We are going to see here, that is the coagulation of blood. So this is initiated by the injured cells. So the injured cells or the tissues will produce a substance called as the thromboplastin. And this thromboplastin will attract the platelets. And this platelets will also produce more thromboplastin. And what happens here is, this thromboplastin lead to the production of an complex enzyme called a thrombokinase. And this thrombokinase is produced as a result of a reaction called as cascade process, and this is an enzyme linked reaction. Okay, so because of this cascade process, thrombokinase is produced. Now what happens? These thrombokinase will be activating the prothrombin, which is found in an inactive form in our body, into thrombin. So now these thrombins are active, and this will take place only in the presence of calcium. And this thrombin, what thrombin will do? Thrombin will be activating the inactive fibrinogen present in our body into the active form, that is fibrin. This is also occurs in the presence of calcium. So these fibrins, along with the dead formed elements, will form the clot or coagulum. So this is how the blood coagulation is taking place. So next we are going to learn blood grouping. So there are two types of blood grouping. One is A, B, O blood grouping, and the next one is RH grouping. A and B are different types of antigens present on RBC, and based on the presence or absence of these antigens, blood group is of four types. That is A, B, and O. So based on the presence of antigen, A blood group people have antigen A in their body, B blood group have antigen B, and AB blood group have both antigen A and antigen B in their body, and the O blood group people do not have any antigens on their RBC. And there are antibodies also present in the blood plasma, and these antibodies for blood group A is anti-B, blood group B is anti-A, and AB blood group people, since they have both the type of antigens, they won't be having any antibody in their body. Now coming to the O blood group people, since they don't have any antigen present in them, they have both anti-A and anti-B antibodies present in them. So what you can understand from this is, we cannot mix and match the blood. So let us take blood group A. So a person with a blood group A can take blood from A blood group and can also accept blood from blood group O. But they can only donate blood to blood group A and AB. Same way, blood group B. Blood group B can take blood from B and O, but can only give blood to B and AB. Now coming to the blood group O or AB. AB blood group people can take blood from O, A, B and AB. But they can only give blood to AB blood group people. They cannot give blood to anyone else. Coming to O blood group. O blood group person can give blood to A, B, AB and O. But they can only take blood from O blood group people. Now we can see what is RH factor. Now we will see what is RH factor. This RH factor is a type of antigen present on the surface of RBC. 80% of people are having RH antibody and they are called as RH positive, and 20% of the people do not have this RH antibody on their RBC, so they are called as RH negative. And what is RH? RH is rhesus. And why do they have this RH factor or rhesus factor? Because this factor, there is a similar factor present in rhesus monkey. Now we will see a special case of RH incompatibility, that is erythroblastosis fetalis. This condition only occurs when the mother is RH negative and the fetus is RH positive. So if an RH negative mother is pregnant with an RH positive fetus during the first pregnancy, no problem will be there. So the placenta is there which properly separates the mother's blood and the fetus blood. But when the delivery occurs or during parturition, there is a chance of mixing of this mother's and fetus blood. So a small amount of blood will be mixed in the body. So what happens here is, the RH positive antigen which entered into the RH negative mother, the RH negative mother's body will produce an RH antigen, and this RH antigen will fight against the fetus cells and will destroy it. But afterwards, in the second pregnancy, what happens? This was a primary immune response which was created by our body, and that was a very mild response. Now when the mother is next time pregnant with an RH positive child, what will happen? Our body, our memory cells which are produced in our body will detect the RH antigen and they will cross the placenta, go to the fetus and either destroy the fetus or it will cause severe jaundice or anemia in the growing fetus. So how to prevent this is to take an anti-RH antibody immediately after the first delivery. So this can prevent the erythroblastosis fetalis. Now let us see what is lymph. Lymph is a colorless fluid with specialized leukocytes, and these helps in immune responses in our body. So when the blood is flowing through our body, some amount of blood will be squeezing out, that is the water along with the water soluble substances will be squeezing out from the blood vessels and it will come to the interstitial space. And this is called as interstitial fluid or tissue fluid. And this interstitial fluid has to be taken back to the blood so that the blood volume can be maintained. And this is done by the lymphatic system. So the lymphatic veins and capillaries will be collecting back the tissue fluid and it will be draining it back into a larger vein. And why it is drained into a large vein? Because once the tissue fluid is there, it will be exchanging gases and the oxygen will be removed, carbon dioxide will be taken in. So now this is deoxygenated and now this has to be given to the vein so that it will be taken to heart for pumping into lungs for oxygenation. Lymph is also an important carrier of hormones as well as nutrients. The fats are absorbed from our digestive system through the lacteals which are present in the villi and then later given to the blood. We will be discussing three types of circulatory pathways here. The one which is present in fish, The second one is present in amphibians and reptiles. And the third one, which is found in aves and mammals. Fish is having a two-chambered heart. So two-chambered heart means that is one auricle will be there and a ventricle will be there. Okay. Reptiles are having three-chambered heart. So the amphibians and reptiles are having a three-chambered heart. That is, they have two auricles and one ventricle. Now coming to the aves and mammals, they have four-chambered hearts. And that is, they have two auricles and two ventricles. And now we will see how the circulation is taking place in fish. So fish heart is a venous heart. It is venous. Why? Because this heart only receives deoxygenated blood. Heart will be pumping out deoxygenated blood. So heart will be pumping out deoxygenated blood, and this deoxygenated blood will be going into the gills. And where what happens? Where the blood will be oxygenated and it will be taken to different body parts. So different parts of the body. And what happens there? That oxygen is given. And then it becomes deoxygenated. Again go back to the heart and then just pumped back into the gills for oxygenation. So they have only one type of circulation. Only deoxygenated blood is coming into the heart of the fish. So this type of circulation is called as single circulation. Now whereas in reptiles and amphibians, what will happen? The atria will be receiving blood separately. That is, the right atria will receive deoxygenated blood and left atria will be receiving oxygenated blood. But these two blood will be getting mixed in the ventricles. This type of circulation is called as the incomplete double circulation. Aves and mammals have complete double circulation. Now we will see different types of circulatory system. So one is open circulatory system and the other one is closed circulatory system. So open circulatory system is mainly found in mollusca and arthropoda, whereas closed circulation is found in annelida and chordata. So in open circulatory system, what will happen? Here heart is there and this heart will be pumping out blood through large vessels and the blood will be flowing in the body spaces called as the sinuses. And coming to closed circulatory system, heart is there and the heart will be pumping out blood through definite arteries, veins and capillaries through a closed network of capillaries or arteries and veins. Now next we are going to learn the human circulatory system. So human circulatory system consists of the heart, blood vessels and the blood. So we have already finished our blood. Now we will see human heart and blood vessels. So first we will see blood vessels. So the blood vessels includes arteries, veins and capillaries. So first we will see the structure of arteries, veins and capillaries. The inner layer is called as the tunica intima. The middle layer is called as the tunica media. And the outer layer is called as the tunica externa. The tunica externa is made up of collagen fibers. So this is made up of collagen fibers. And the middle layer is made up of smooth muscles. And the inner layer is made up of simple squamous epithelium. And the inner portion of these arteries and veins are called as the lumen. And lumen of your veins have valves present to prevent backflow of blood. Now we will see the structure of human heart. So you can see here, heart is made up of 4 chambers: the right atrium, right ventricles, left atrium and left ventricles. The septum between two atriums are called as the interatrial septum, and the septums between the ventricles are called as the interventricular septum. The septum between atrium and ventricles are called as the atrioventricular septum. So in the atrioventricular septum, we have valves present, okay, and the one which is present in the right atrioventricular septum is called as the tricuspid valve, and the one which is present in the left atrioventricular valve is called as the bicuspid valve or mitral valve. So from different parts of the body, veins and venules will collect deoxygenated blood and will give to vena cava, and this vena cava drains deoxygenated blood into the right atrium. So from the right atrium through the tricuspid valve, the blood will reach the ventricles. So from the ventricles, the pulmonary artery is taking blood to lungs for oxygenation through the pulmonary semilunar valve. And now once the blood reaches the lungs, what happens? The blood will get oxygenated and that will be taken back to heart through pulmonary vein, and this pulmonary vein drains blood into the left atrium. From the left atrium, blood moves to the left ventricles through the bicuspid valve, and from the bicuspid valve, blood is pumped out to different parts of the body through aorta, and that is through the aortic semilunar valve. Now, the upper right corner of right atrium contains a nodal musculature called as SA node, which is capable of producing its own action potential, and this leads to the contraction of atrial muscles as well as the heart. And there is another tissue which is present in the lower left corner of right atrium called as the atrioventricular node. So this atrioventricular node receives signals from the SA node. And right from the atrioventricular node, a bundle of nerves arises into the atrioventricular septum called as the atrioventricular bundle or AV bundle. So once the AV bundle reaches the ventricular septum, it will suddenly divide into two bundle of his or bundle branches, and this bundle branches enters into the ventricular walls and is called as the Purkinje fibers. So these Purkinje fibers spread throughout the ventricular walls. So there are certain ligaments present in the heart which connects the valves to the ventricular valves. This is called as the chordae tendineae. So this is all about the structure of human heart. So the nodal musculature, which is called as the SA node, which is present on the upper right corner of right atrium, can produce its own action potential, and it can produce approximately 70 to 75 action potentials per minute. And this is responsible for maintaining the contractile activity of heart. And this SA node is also called as the natural pacemaker of heart. So what does that mean? So what does this mean? Our heart can beat 70 to 75 times in a minute, or per minute our heart can beat 70 to 75 times, and that is an average of 72 beats per minute, which is known as the heart rate. Next we move on to the cardiac cycle. Cardiac cycle includes three events. One is the joint diastole, then you have the atrial systole, then followed by the ventricular systole. So we'll see what is joint diastole. So during joint diastole, atrial filling will take place, that is both vena cava as well as pulmonary artery will fill the atrium, that is atrial filling. So at this time, the AV valves, what are AV valves? The tricuspid and bicuspid valve, which is present in the atrioventricular septum, is called as the AV valves. The AV valves will be open and the semilunar valves will be closed. Okay, so since the AV valve is open, partial ventricular filling will take place. So 70% of the ventricle will get filled at the time of joint diastole. Now what will happen at the time of atrial systole? So during atrial systole, SA node produces action potential. So SA node will produce action potential. So this action potential leads to the contraction of atria. So atria will contract and this leads to the complete filling of ventricles. So complete ventricular filling. So rest of what 30% was left out will be pumped out to the ventricles. Now here AV valves are all open and S semilunar valves are closed. So now what will happen during ventricular systole? So what will happen during ventricular systole? So whatever impulse which is produced by the SA node will reach the AV node now. So the AV node will give signal to AV bundle. AV bundle gives it to bundle of His, and from bundle of His it goes to Purkinje fibers. So during ventricular systole what will happen? The blood from both the ventricles will pumped out to different parts of the body through aorta. And so what happens during ventricular systole? So the oxygenated blood from left ventricles is pumped out through aorta to different parts of the body. Then again what will happen? From the deoxygenated blood in the right ventricle will move through pulmonary artery to lungs for oxygenation. Here both AV valves are closed and the semilunar valves are open. So these are the events that are taking place during the ventricular systole. Now we will see what is lub and what is dub. So lub is the first heart sound and dub is the second heart sound. So first heart sound is heard when AV valves are closing. So AV valves closure will produce the lub sound, and closure of semilunar valves. So semilunar valve closure produces the dub sound. So when will this AV valve close? The AV valve close in the beginning of ventricular systole. So this happens in the beginning of ventricular systole. And when will the semilunar valve close? So once the ventricle contracted, the blood flowed through a pulmonary artery and aorta. So now what will happen? It should prevent the backflow of blood into ventricles, right? So the beginning of ventricular diastole, semilunar valve will be closed. So when will semilunar valve close? Beginning of ventricular diastole. Now let us see how to calculate the cardiac output. So what is the heart rate? So heart can beat 72 beats per minute. Okay, so per minute heart can beat 72 times. And what is stroke volume? It is the amount of blood which can be pumped out by each ventricles during a contraction or during a cardiac cycle. So what is stroke volume? Stroke volume is approximately like 70 ml. So how can we calculate the cardiac output? So cardiac output is the product of heart rate and stroke volume. So cardiac output is 72 into 70, that is approximately equal to 5000 ml, that is approximately 5 liters. So this is the cardiac output. So that is the amount of blood pumped out from our heart in a minute. So next we are going to see what is ECG, electrocardiogram. It is a representation of electrical activity of our heart. It's a graphical representation. And this is measured by an instrument called as electrocardiograph. So this is the device which is used to measure the, or instrument which is used to measure the ECG or electrocardiogram. These leads are connected, 2 to the wrist of hand and 1 to the left leg angle. Now we'll see explanation of the graphical representation. So what the P wave here represents is atrial depolarization. That is the atrial systole. The QRS complex represents the ventricular depolarization or ventricular systole. Now what does T wave represents? It represents ventricular repolarization, or it is called as the ventricular diastole. So what does this Q represents? Q represents the initiation of ventricular desystole, and the S is the place where ventricular contraction or systole is completed. Now we are going to learn what is double circulation. So you can see here we have a heart, then you have the lungs, and the body tissues are present. So here from different parts of the body, deoxygenated blood is carried to the right atrium. So this is done by the vena cava. Okay, so from the right atrium, blood will move to the right ventricles. Now the deoxygenated blood from the right ventricles is taken to lungs for oxygenation by the pulmonary artery. So once the blood reaches the lungs, what will happen? It will get oxygenated and it is taken back into the heart, that is to the left atrium, by the pulmonary vein, and that is oxygenated blood. Now this oxygenated blood will move to the left ventricles, and from left ventricle through aorta, it is taken to different parts of the body. So this is what is called double circulation. And why is it called double circulation? Because it contains two circuits or two circular pathway. One is systemic pathway and the other one is pulmonary pathway. And also blood will reach into heart two times, one as oxygenated and one as deoxygenated. That is why circulation in four-chambered heart or circulation in That is why this type of circulation is called as double circulation, and this type of circulation is achieved by all the animals with the four-chambered heart. And in this, mixing of oxygenated and deoxygenated blood will not take place. Now we will see the pathway of systemic circulation and pulmonary circulation. So systemic circulation is from the left ventricles. Oxygenated blood is taken through the aorta to different parts of the bodies like tissues, and from there deoxygenated blood will be taken through, superior and inferior, and from there deoxygenated blood will be taken through to vena cava and to the right atrium. So pulmonary circulation. So from the right ventricles, deoxygenated blood will be taken through the pulmonary artery to the lungs for oxygenation, and after oxygenation, pulmonary vein will be taking it back into the heart to the left atrium. So this is the pulmonary circulation, and this pathway is important. Next we will see the regulation of cardiac activity. Human heart is regulated intrinsically, that is auto-regulated by nodal musculatures, and this nodal musculature is the SA node, and thus our heart is called as myogenic heart. Because it is controlled by specialized muscles, it is called as myogenic heart. Now, a region in the medulla oblongata also controls the activities of heart. So that is by the autonomous nervous system. So two parts of autonomous nervous system includes the sympathetic nervous system and parasympathetic nervous system. So the sympathetic nervous system will increase the heart rate, and the parasympathetic nervous system will decrease the heart rate. And the adrenal medullary hormones also increases the heart rate. So what are the adrenal medullary hormones? Adrenaline and noradrenaline, which will increase the heart rate as their emergency hormones, right? Now moving on to the disorders related to circulatory system. So the first one is hypertension. It is nothing but the high blood pressure. So what is blood pressure? Blood pressure is the pressure exerted by blood onto the walls of arteries. So what is the normal blood pressure? Normal blood pressure is 120 by 80 mmHg. So what is 120? It is the systolic pressure, and what is 80? It is the diastolic pressure. So on repeated checks of blood pressure, if the blood pressure is always above 140/90 mm per Hg, that condition is known as hypertension or high blood pressure. Next is coronary artery disease, and this is also called as atherosclerosis. So this is the condition when the coronary artery is getting blocked by calcium, fat, cholesterol or fibrous tissue. So what is coronary artery? It is the artery which takes blood to the muscles of heart. So when these are blocked by like calcium, fat, cholesterol and fibrous tissue, what happens? There is no sufficient amount of blood reaching the heart muscles. Next is angina. It is also called as angina pectoris. So this is the condition when no sufficient oxygen is reaching the heart muscles. This can occur in male and female in all the age groups, but mainly found in elderly people. Next is heart failure. This is the condition when our heart is not able to pump out blood sufficiently to meet the requirements of our body. It is also called as congestive heart failure. Cardiac arrest and heart attack is not heart failure. Cardiac arrest is sudden stopping of heart. Heart attack is when the heart muscles are getting suddenly damaged because of inadequate supply of oxygen. So these two are not same as that of heart failure. Heart attack is also called as myocardial infarction. So by that we completed body fluids and circulation chapter. So if you have any doubts, you can ask me in comment section or you can send me personal messages. If you like my class, like and share it, and if you have not subscribed yet, subscribe it so that you won't miss the future classes. Okay, thank you. Have a nice day.